All right, so it is now time for us to start talking about Mendelian patterns or patterns of inheritance. So, like taking a trait and passing it down from parents to offspring. And this is kind of the time of the year where you students often like to think that you remember enough of this to kind of get by. And so, I'm just going to caution to you that what you learned, in freshman, sophomore year of high school or in middle school is a good primer for what we're going to be covering in this unit for AP Bio, but is not at all a replacement for it. So this man's face might look a little familiar to you. This is Gregor Mendel. He is one of the most famous scientists of all time. He was actually an Austrian monk, and we consider him to be the father of genetics. He worked with what is called pea plants, so plants that make peas, right? And he actually tracked a bunch of different traits on there, and we're going to talk about a few of them. So for now, we're just going to look at seed shape, seed color, flower color, pod shape, pod color, stem height, and flower position. These are 7 different traits that he tracked within a population based on what parents an individual had and what children they might have, etc. So he was basically interested in studying traits and how they get passed down. So seeds can either be round or wrinkled. They can be yellow or green. Flowers can be either purple or white, etc., etc. There's 2 different options for all of these 7 traits. So what he would do is he would cross a bunch of peas together. So cross basically just means you like make them mate. You force them to mate. Okay. And he would get what we call pure breeding individuals. So this individual right here, this green plant at the top, that's a pure breeding green plant, which basically means it only has green in it. This one right here, that's going to be a pure breeding yellow. So that means it only has yellow. Think about it like a dog. If you have like a purebred golden retriever, it has only ever had golden retriever in it. There's no Doberman parents or anything like that. So what we have going on right here, this is a cross happening. He's basically taken the green parent and the yellow parent, and he's crossed them, and so now he's making an F1 generation. So we typically will call this the P generation for parental, and then we have an F1 generation, and then we have an F2 generation. So the F1 generation in this case was all yellow. Okay? So even though there was a green parent, they were all yellow at the end. Okay? So that should tell you something. We're going to talk about what that means. But then if you take the F1 generation and you cross it with itself, okay, so you take one individual from this generation and another individual from this generation, and you cross them, their children will on average be 3 out of 4 times will be yellow, and 1 out of 4 times will be green, so 25%. So that ratio right there, 1 to 4, or 1 to 3 rather, 1 out of 4, right, is probably bringing back to you some ideas about this thing that everybody loves because they're really, really easy. Well, they're not all easy. These are Punnett squares. Okay? They're not all easy, and we're going to talk about why. But I just want you to kind of start to think about them and like how we might begin using them because they probably look familiar to you. So just a little bit of a throwback to meiosis. Okay? Just a reminder that meiosis creates haploid gametes. So haploid means you have 1 copy of every chromosome. Gametes are sex cells, so sperm and egg. And basically that just means that they have half as many chromosomes as their parents did. Okay? So your egg in a normal human egg should have 23 chromosomes. Your sperm in a normal human sperm should have 23. You combine that and you get 46 in the baby or the zygote. Okay? That's going to be the basis for what we are doing with Gregor Mendel's plants because this is the basis of reproduction. And so if we're going to be tracking how traits move across reproduction, it's important to just remember where this information comes from. It comes from the chromosomes. Okay? So during gamete production, so that sounds really fancy. That's just meiosis. Okay? Gamete production is just the making of gametes, so meiosis. Okay? Excuse me. There's a term here that you have hopefully recalled from the past. It's alleles. So alleles are basically versions of a gene. Okay? And so if we look back to this table right here, if there are 2 different kinds, like there's a yellow and there's a green, that means that there's a yellow allele and there's a green allele. Okay? If there's purple and there's white, that means that there's a purple allele and a white allele. So this is when you start to see that like capital P and lowercase p kind of situation that you might be useful, or capital R, lowercase r. Okay, those are alleles. Okay, so the letters are alleles. And those alleles are just part of the chromosome. Like they're determined by the chromosome. It's just information that's in the chromosome that determines what kind of a P shape you're going to make, or what kind of a hair color you're going to have, or whatever. Okay? All of your traits almost are determined by genetics, or a combination of genetics and environment. So what we have here in the top is we have meiosis 1 occurring. So this right here, this blue bar is metaphase 1. Okay? So that means that these arrows here are going to be representing that prophase to metaphase. Okay? So, excuse me. Here I have two different chromosomes. I'm going to call it 1A for this Y, this capital Y here, 1B for this lowercase y. I have 2A for the lowercase r and 2B for the uppercase r. That part doesn't matter. Okay? You're going to start to see terms like heterozygous. Okay? So hopefully you remember that hetero means different. And basically that just means that your alleles don't match. So like I have 2 copies of every chromosome, right? That should be a review. I have 2 copies of every chromosome. And if I have an allele on every chromosome, then that would mean that like if I'm talking about seed shape, I could be a big R. That could be my genotype. Or I could be a big R, little r, or I could be a little r. Okay? So those right there, those are all genotypes. Okay, and we're going to be practicing with that. These are all genotypes. Okay, and basically, if yours don't match, so you see how this one in the middle has a big R and a little r, those aren't the same thing. So those are different. So this is a heterozygote. Okay. This one over here has 2 capital R's. Those are the same. So that's a homozygote. And over here, this little r, those are the same. So that's a homozygote. So just to kind of prime you on some of the vocab that we're going to need to practice with. It is a lot, but most of it you've seen before. So just as a quick little visual, here I have a bunch of different combinations. Here I have capital B, capital B. So just notice that the alleles are just one little part of the chromosome. Okay? And that's just because these chromosomes are made up of genes, and this gene has different versions, and this version happens to be capital B, that version happens to be capital B. Okay? This individual over here has a capital B and a lowercase b. This individual has a lowercase b and a capital B. The order doesn't matter. Generally, you're going to write it like this with the capital first. Okay? You will rarely write it with the lowercase first. It's never wrong, but it might confuse you. So at least be consistent, whatever you do. And then you can also be homozygous for the little b, so having 2 of the little b. So take a quick second and practice whether these things are heterozygous or homozygous. So I would recommend that you pause the video here, and then I'm just going to go through a few of them. So this guy up here has 2 capital A's, so that's homozygous. This guy right here, capital B, little b, those are different, so that's heterozygous. And then so on and so on. So capital C, little c, well, those are different, so that's hetero. And then big D, well, those are the same, so homozygous, etc. Okay? So you should be able to do that very quickly. That should be basically immediate. No fancy stuff there. Okay? So these 2 terms are so important, and it is so important that you learn them and you use them correctly, especially in like our FRQs, for instance. Here we have genotype. So I used that term earlier. This is the combination of alleles an organism has. That sounds really, really fancy and really confusing. It's not. Okay, so remember when I had a little r, or excuse me, a big R, little r? All right, that's a genotype. Okay, that's what my genotype would look like. It's determined by your chromosomes right here. Okay, so if I were to track a gene, maybe it's this bottom one down here. This one says big R, this one says little r. Okay, the phenotype is the physical trait. So the pH in phenotype is typically equated to the pH in physical, just to kind of help you remember. So phenotype is physical, genotype is genetic. Okay, so your genetics code for your phenotype. So whatever your DNA says is going to determine your phenotype. So that begs the question, if we can have homozygote individuals or heterozygote individuals, how can we start to figure out somebody's phenotype based on their genotype? So what I have here are 3 genotypes. You know they're genotypes because they're just letters. And right above them, I have their phenotypes. So this is one of the traits that Mendel himself tracked. Okay, he has purple flowers versus white flowers. And what you should notice is that if you have a purple allele, so capital A is purple, okay? So if you have a purple allele on one chromosome and a purple allele on another, guess what? Shocker, you're going to be purple, okay? If you have a white, so white is little a in this case, if you have a white allele on one chromosome and a white allele on the other, you're going to have, you guessed it, white flowers. Okay, those two are not shocking. But then it begs the question, okay, what happens if I have both? What happens if I am a heterozygote? So that's what this is representing right here. That's a heterozygote. Okay, now in this case, I will tell you that if you are a heterozygote, you will have purple flowers. Okay, so it's almost like we're ignoring this white flower allele. Okay, and that's because capital A, or purple, is dominant to the recessive little a, or white allele. Okay, so basically you can only have white flowers if you have nothing else, okay? If you have no purples. Okay? So that's why the only option we have for white flowers is this case over here on the right, where there are no capital A's, there are no purple alleles. If there's purple, it's going to get expressed. That's what it means to be dominant. If you have it, you show it. Okay, recessive alleles, on the other hand, those don't always show. You might have the allele, but not actually show it. So this individual in the middle, this heterozygote, has the allele. He has a little a, but it doesn't show in his phenotype, right? His color of his flowers is all purple. Okay. So, excuse me. This is just for a little practice. So go ahead and pause the video and start to figure out the phenotypes of individuals with different genotypes considering this information. Okay, so I will do 1 or 2 for you, or with you, I guess. Let's pretend like you're doing this. Hopefully you are. So in this top case, we have purple flowers being dominant to white. So this is exactly like what we saw before. And then on the right side, we have brown eyes being dominant to blue eyes. So if purple is dominant to white, and I have big P and I have little p, that means that my big P is the dominant one, so that is purple. We always put dominant as being capital. Okay, and then white is our recessive, so I'm going to put it right here, white. So now I just need to figure out what my phenotypes will be. Well, if I'm big P, excuse me, I'm going to be purple. Okay, perp. Okay, and then if I'm little p, well, I'm white, so I'm going to be, you guessed it, white. But if I'm a heterozygote, I need to be whichever one is dominant. So in this case, that's purple. Okay. So let's go ahead and do this brown eyes one. So brown eyes is dominant to blue eyes. Okay. So that means that capital B is the dominant one, as always. So capital B is going to be brown. Lowercase b is going to be blonde, or excuse me, blue. Just thinking hair for some reason. So if I'm brown, you guessed it, I'm brown. If I'm blue, you guessed it, I'm blue. But the heterozygote, the individual who has one of each, is going to be whichever one's dominant, which in this case is brown. Okay, so this you should be able to do rather quickly once you kind of practice it. So now that we have an idea of what genotypes are, we need to start thinking about gametes. So remember that gametes are sex cells, they're sperm and egg. And individuals make these, and obviously they hold your genetic information, right? If you make sperm or if you make eggs, that sperm or egg contains your chromosomes, right? We saw that in meiosis. So what we need to do is start thinking about the alleles that might show up inside of these gametes. Okay, so right here I have the symbol for a male, and right here I have the symbol for a female. You don't need to worry about pollen and pistil. Those are just different parts. Well, the pistil is a part of a flower, and the pollen is released by the flower. It's basically just sperm and egg. Okay, so just picture it as sperm and egg. It just makes things simpler. Okay, what you will see here is that this individual on the top, this male, this dad, is passing either a capital B or a lowercase b. Okay, what's the only situation that he could be? What could his genotype be if he's passing both of those? Okay, if he could pass both, that means he had to have both. So he's a heterozygote. He's a capital B, little b. Okay, mom over here can pass a big B or a little b. So her genotype is also big B, little b. Okay, so the parents are both heterozygotes. Okay, now this Punnett square, everybody loves Punnett squares, and it's because once you figure out what's on the outside, you literally just have to fill in the inside, and that's great. That's really useful, but you need to understand what it is. Otherwise, it's going to be completely useless to you, both on my tests and on the AP test. So, if dad is a big B, little b, he can either pass a big B or a little b, and he has an equal chance of passing each. So that's why we have this like breakdown over here. We have two columns. Okay, it's basically like, hey, 50% of the time I'm going to do that, and 50% of the time I'm going to do that. Mom's the same way. 50% of the time I'm going to pass a big B, 50% of the time I'm going to pass a little b. Okay, so if dad up here passes a big B, that happens half the time. Okay, and mom passes a big B, that'll happen half the time, then that means that my child will have a genotype of big B, big B. Okay, so this half thing has to happen, and this half chance has to happen. So if they both have to happen, you multiply them. So 1/2 times 1/2, well, that's just 1/4. So a quarter of the time your kid is going to look like this. It's going to have that genotype. Okay, 50% of the time, though, dad is going to pass a little b, and mom will pass a big B. Those are both possible. And in fact, they're equally as likely because dad has a half chance of passing little b. Mom has a half chance of passing big B, just like they had last time. So this is going to be another quarter. Okay? And this is why there's 4 boxes. Each one is equally likely. If this were to happen, the individual would be heterozygous. They'd be big B, little b. But the opposite could happen. Dad could pass big B, mom could pass little b, and then we would have this child down here. That's a quarter chance. And the only way you can have a white child with the flowers, okay, is if mom passes a little b and dad passes a little b. Okay, that's the only way we're going to get a white flower child. So that means that both of those things have to happen. They're both 50% chance. So 50 times 50, or excuse me, 1/2 times 1/2 leads to 1/4. So a quarter of the time you're going to have white flowers in your children between these two parents. Okay, so there are 3 times as many purple flowered children as there are white children, and that is going to become very, very important. And it kind of takes us back to the green versus yellow. Basically, we have this ratio, 3 yellow for every 1 green. Well, that's a really good tell that this individual is a heterozygote and that individual is a heterozygote. Okay, so you're just need to get to practice with Punnett squares, and we're going to do a lot in class. So now you need to kind of work backwards. I give you a Punnett square. You need to be able to figure out the genotypes of the parents. Okay, and so you're going to write them in a cross form right here. And then you need to be able to fill in the offspring's genotype. So go ahead and pause it and just on a piece of scratch paper, figure out the genotypes of the parents and the genotypes of the potential offspring. So up here I have parent 1. This is parent 1. He can pass, or she. Typically I'll put the male on top, but it doesn't really matter. He will pass a big R, or he can pass a little r. So if they can both happen, they're both equally likely, then, and I only have 2 alleles, right? So then what's my only genotype possible? Well, that means that I have to have one of each. So I'm a heterozygote. I'm a big R, little r. Okay. This individual can pass a big R or they can pass a little r. So same thing. Okay. And then you just need to fill in the Punnett square. You guys have done this a million times. I'm sure this is what everybody loves from bio because it's easy. You just put the column and the row from each one. Now, what you'll notice is that I always put the capital one first, and I mentioned this earlier, but I just always like to keep it consistent because it's really easy for me to be like, okay, that's going to be round or purple or whatever it is that you're tracking because it has big R. This one's going to be round. This one's going to be round. Oh, this one's going to be wrinkled or whatever it is that you're tracking. Okay, because there's no dominant one out front. Okay, so that's how you fill in a Punnett square. And then you need to figure out like, okay, what does that actually mean? So a quarter of the time my kid is going to be a big R, quarter of the time it'll be a little r, and half the time it's going to be a heterozygote. So a big R, little r. And all three of these will look the same as far as what we've seen so far. Okay, so you should have a 3-to-1 ratio if it's one of these traits. Okay. And we can do this with lots of different things, and Mendel did this with a lot of different things. So he did this with pea plant color, or excuse me, the pod color. So you could be yellow or you could be green, and you can figure out right now which one is dominant and which one is recessive, not just because I've written it, but because if you look, you just have to find a heterozygote. So anybody who has one of each, these guys both do. What is their phenotype? Oh, they're yellow. So that means that yellow is dominant. Okay. And we're going to practice with that. You can do the same thing with brown eyes. So just take a quick second to kind of analyze this Punnett square, figure out what it's trying to tell you. All right. So what we saw in both of those Punnett squares was really, really nice and simple. It was just a single trait. We only cared about pod color, or we only cared about eye color. And that's great. That's so easy. Unfortunately, that's not what we have to limit ourselves to because you have millions of traits in your body. And so we need to kind of put them together sometimes. We need to look at multiple. So what we have are what we call dihybrid crosses. So di means two. Hybrid basically means that they are a combination, and I'll talk about that in a second. So a monohybrid cross is what we saw before. That's when you just do one trait. A dihybrid cross is when you're going to do two traits. Okay. And so what you need to have then are two different letters. You need to have the letter G for one trait. So that's going to determine whether you're green or you're yellow. Okay. And we need to have the letter R for whether you're round or wrinkled. So that would be like your shape. Okay. So there's two traits. There's color and there's shape. We're tracking two traits at once. So now when you have a genotype, you're going to start to have four letters because we have one gene, two genes, one trait, two traits. Okay. So it gets really, really complicated because when you have an individual who's heterozygous for both, so like this individual right here, let's just focus on this one. Okay. He could pass a big G, right? He has a big G, so he could pass a big G. So he could pass a big G. Now, he's only going to pass one G at a time. But with that G, could he pass a big R? Yeah, he has a big R. He could pass a big R. Okay, so that's one possible gamete. So could he pass a big G but with a little r? He could. Could he pass a little g? Is that possible? Well, does he have one? Yes. Okay, well then yes, it's possible. So with that little g, could he have a big R? And with that little g, could he have a little r? So there are four possible gametes right here for this one individual. Okay, meaning that that's a sperm, that's a sperm, that's a sperm, or that's a sperm. Any of them are possible. And then this individual, they have the same genotype, so they can make actually the same exact children. Or excuse me, I should say gametes. They can make the exact same gametes. Okay? So that means that you have to figure out a Punnett square that's like got four possibilities on the top and four possibilities on the side. Okay. And then you would fill in this Punnett square, and obviously it becomes a giant disaster. But it's going to be a 4x4 grid, 16 boxes. Okay, so this parent right here on the left can pass a big G with a big R, or they can pass a big G with a little r, or they can pass a little g with a big R, or a little g with a little r. And then this parent can do the same thing over here. So then you'd have to fill in every possibility. Okay, this would be a dihybrid cross. It is challenging your first time. Okay, but it really just comes down to statistics. If I'm going to have this combination a quarter of the time and that combination a quarter of the time, well, if both of those happen, I'm going to have this child, and that'll happen 1/16 of the time. Okay? And you can fill all of these in like this, and it's a really, really good exercise. Once you set up the columns, though, it's the same thing as a normal Punnett square, it's just writing them in. So really the main practice comes in with making the columns, figuring out what gametes an individual can make. So that is the thing that you need to practice more than anything. Filling in the Punnett square is easy. Anybody can do that. You could probably do that walking in today. But a lot of people can't do the columns and rows. And that's what you need to be able to do. So once you do this, once you kind of get everybody's genotype in there, then you have to figure out a phenotypic ratio. So we have a genotype and we have a phenotype. Remember, phenotype is physical. Okay. So now we have to remember, like, oh yeah, these G's and R's and things mean something. Okay. So the R was round, the capital R was round. Okay. So I'm just going to write that right here. So capital R is round. Okay. Little r was wrinkled. Okay. Big G was green. And little g was yellow, I believe. Okay. So this individual right here has two big G's. So that means they have two greens and they have two big R's, which means they have two rounds. So this person is going to be green and they're going to be round. Okay, that's what their P's are going to look like. This one's going to be green and there's a heterozygote. Well, which one's dominant? Oh, the capital one. So they're going to be round. Okay, so they're going to be G for green and they're going to have round seeds. So those two individuals have the same phenotype. Okay. This individual way down at the bottom right, if I can find my little cursor here, I always lose track. Down here in the bottom right has two little g's. So little g's are for yellow. Okay. And two little r's, well, little r's are wrinkled. So this individual is going to be yellow and wrinkled. Okay, so entirely different from the one up there. And so if you went through and you labeled everyone, you would see a 9:3 to 3:1 ratio. You would see 9 green and round, like we saw in the first two. You would see only 1 that's yellow and wrinkled. That's what I saw down here. Okay. And then you would see 3 that were yellow, but they were round. So that would be like this individual. They have to have 2 little g's to be yellow, and they would have a capital R to be round. Okay. And then you would have 3 individuals that are green and wrinkled. So they would have to have a big G. Big G, and they would have to have 2 little r's to be wrinkled because it's recessive. Okay? And so you just need to practice understanding what is happening in these Punnett scores and why they matter. It's just statistics, it's just likelihood, it's just probability. Okay? And so you have to learn to use it, and it is challenging. All right. So when you start to use Punnett scores effectively, you can kind of figure out what's going on with different traits. You can try to figure out like whether this is a single trait, like a monohybrid cross. So like if 3 times as many people have brown hair as blonde hair, that kind of should tell you like, oh, okay, brown is probably dominant to blonde. But if you see a 9:3 to 3:1 ratio, that should tell you, oh, there's 2 traits here that we should be keeping track of. Okay, and there's more to it than that, and we are going to practice it, but it's a lot. Okay, and so here's some videos that might help you. Okay, there's a TED Ed talk about Mendel's history. There's a crash course on heredity that I always recommend. And then for Punnett squares, especially if you haven't seen them, there's a great Bozeman video. Okay, and then this kind of takes me back to Mendel. Mendel came up with 2 laws. Remember, laws are like the ultimate thing in science. It's like you have a hypothesis, that's great. You have a theory, okay, that's really good, because it means it's been tested and it hasn't been disproven. A law is always going to be the case. Okay, so these are basically what we have determined to follow or that traits follow. These are the rules that traits follow. Now, after Mendel's time, we have found exceptions to this rule. And so these, I will say, aren't comprehensive laws. Okay? There are exceptions. These are Mendel's laws, and Mendel was around, in the 1800s, so he has a little bit different understanding of the world. Okay, but excuse me, it is a really important foundation for what we understand now about genetics. So basically, his law of segregation states that there are alleles for given genes, right? We know that. You have two copies or two different alleles, or it could be the same allele. You have two alleles for any trait, and they're going to go into different gametes. So like when you start to make your sperm and egg, if you look up here, I had a big R and I had a little r. Well, those aren't going to end up in the same egg ever, or the same sperm ever. Every sperm should have only a big R or a little r. Okay? And that's what's happening here. Same with the Y here. Every gamete, every sperm, every egg should only get one R and one Y. Okay. The law of independent assortment basically just says that, hey, your little r can combine with a big Y, or your big Y could combine with a big R. It doesn't matter. Okay. So just because you passed this little r doesn't mean you had to pass the big Y. Okay. It'll happen sometimes. That's what happened here. Or excuse me, I meant little y. That's what'll happen here. That'll happen a quarter of the time. But a quarter of the time, you will also pass a little r with a big Y. And a quarter of the time, you're going to pass a big R with a little y, etc. Okay. So your chromosomes are unique. They don't need to follow each other. So just because you get a dominant for one trait doesn't mean you get a dominant for the other trait. Okay. But there are some weird exceptions. We're going to get a chance to kind of talk about that. And don't forget that crossing over happens. So it's not always as simple as you had two little r's, and so that means that you have, you have one big R and one little r. That means you have a 50% chance of passing this and this. Sometimes things kind of get messed up. So we have what are called linked genes. And basically, that means they're less likely to separate during crossing over. And so basically that means that like sometimes a little r will usually go with a little y or something like that. But all of those things are the exceptions to the rule. So I don't want you to get bogged down in that. We are going to practice with those exceptions. Those exceptions are exceptions to Mendel's law, so we call them non-Mendelian genetics, so they don't follow his rules. Okay? Here are 3 videos on Mendel's laws and linked genes that are really, really useful. I highly recommend them. But I just kind of wanted to talk a little bit about disorders, so genetic disorders, and the ethics behind treating them and working with individuals with them. So now that we can track traits, a lot of traits actually are diseases. And so you need to start thinking about what would happen if you had a parent who had a trait such as maybe cystic fibrosis or Down syndrome or any other genetic caused traits, and what that might cause in their gamete production. Okay, and so just to kind of talk about this first, Down syndrome is basically when you get one too many copies of chromosome 21 specifically. Okay, so you're supposed to have 2 copies of every chromosome, but if you have Down syndrome, you have 3 copies. That's why we call it a trisomy of just that one chromosome. And basically it meant that the chromatids didn't separate properly when you were making your sperm or egg. Okay, so meiosis went wrong, mistakes happen. Okay, and so that has huge effects on this individual's phenotype, on their brain development, right? And so it's difficult to talk about genetic diseases without having to kind of tackle the ethics of it, because there are a lot of really awesome tools that might one day be able to help us treat a lot of these diseases. But there are ethics behind it that many people disagree with. So you need to kind of go at it from your own angle, and we will discuss it a little bit productively as a class. I wish we had more time to discuss it because it's really important moving forward. But I just want you to be actively thinking about it and just remembering that people have traits like this. This affects people's lives. That takes me to this. This is a karyotype. okay, or a karyogram. They're the same thing. And it's basically just a picture of somebody's chromosomes. And so normally you would just have 2 of every chromosome. You'd have 2 chromosome 1s, 2 chromosome 2s, etc. That's what this person has. This one's bent, but that's not abnormal. Remember, these things are 3-dimensional. They move around. They do that all the time. Okay, if you were to stretch it out, it would be about the same length. Okay? But if you look down here, they have 3 copies of chromosome 21. So that individual has Down syndrome. Okay? So there are other traits that you need to start thinking about. There are other diseases. Sickle cell anemia is one of the classic ones that you've probably heard of. Basically, it's a recessive disease. So that means that you have to have both recessive alleles to have the trait. Okay? And we'll talk about what that means. But if you do, you have a sickle-shaped blood cell. It kind of looks like a crescent moon red blood cell instead of a normal red blood cell. Okay? And so normally when your blood cells are passing through your capillaries, so those are your really, really thin blood vessels, a normal one can just go right through. But this guy is kind of more jagged. The sickle cell is a little more jagged, and so it might get caught along the way. It might kind of cause a buildup. And so that can cause a lot of different symptoms. But ultimately, you can't carry as much oxygen, so that's why you get fatigued more. Okay? It can clog your arteries. It can cause pain in your joints, and it can eventually lead to the breakdown of some of your blood cells. Your body will kind of have to break itself down a little bit. And I want you to start thinking about genetic diseases in terms of DNA. So I know we haven't like broken down DNA into great detail yet, and that's coming in chapter 16. But remember that DNA is just a string of letters, right? It's C, G, T, and A in some random order. And so those are your chromosomes. So like if I were to take a chromosome and just look at a tiny little bit here, well, it codes for normal hemoglobin if it looks like this. But if you change just a single nucleotide, you change this one right here. It should have been a T. Instead, I have an A. That's a different allele. Okay, so those are 2 different alleles. This one causes sickle cell. This one doesn't. Okay, a single change in a single base pair of a single gene causes a horribly debilitating disease. Okay. So that takes us to cystic fibrosis, and this is another recessive trait, another recessive disease, and this one's on chromosome 7. And basically, if you have 2 copies of the recessive allele, so you're like a little r, or whatever letter they use for it, then that means that you have cystic fibrosis. And I don't need you to know what cystic fibrosis does to your body. That's beyond the scope of this class. It is very interesting, but it is not at all necessary. I just want you to know that we will be tracking traits like this within families, and that's going to be where we're going kind of next, so that we can start to figure out how likely is it that somebody is going to pass that off to their offspring. Okay, so like up here we have, excuse me, we have cystic fibrosis again. It is autosomal, which means it's on any of the chromosomes except X and Y, and we'll talk about what that means. But it's recessive, so that means that you have to have little c to have the trait. Okay, that's the only child out of these 4 that is going to have it. Okay. Now, if you're a carrier father, carrier basically just means that you have it, but you don't show it. So that in this case is going to be a heterozygote, so capital C, little c. Carrier mother, capital C, little c. So mom can pass a big C, she'll do that half the time, and a little c, she'll do that half the time. Dad can do the same. And so a quarter of the time, your child will be a big C, so they'll be completely unaffected. Your child will be a big C, little c a quarter of the time, a big C, little c a quarter of the time again. So these 2 are actually the same. So this is really half the time. So about half of your kids will be carriers. Okay, so they'll have the allele but not show it. And then a quarter of your children will have cystic fibrosis. So that would be this little c, little c. Now, this individual or this family right here shows just probabilities. Okay, it's easy to think that that means that if your first kid is going to be unaffected, so that's this guy right here. Your second kid is going to be big C, little c. And then your third kid is going to be big C. That's not how it works. Okay, your first kid could be little c, little c. Your second kid could also be little c, little c. Okay? Each kid has a quarter chance of this happening to them. It's not like, oh, you got to have an even number, right? That's not how it works. It's the same thing as sex. So when you are having a child and you are determining the child's sex, right? XX is a female, XY is a male individual. Well, half the time, on average, you should get a male child, and half the time you should get a female child. But there are families out there that have 4 male kids and no female kids. That happens all the time. It's just chance. These are just probabilities. Okay? So it's not an absolute rule. And there are a lot of other examples of stuff that I'd like to talk about. Um, and we will get to talk about a lot of normal traits like that are on normal autosomes. But what we're also going to move into, and we're not going to do this right now, but we're going to be moving into traits that are on the X chromosome. So that would be an X-linked trait or an X-linked gene. So for instance, color blindness, red-green color blindness specifically, is actually on the X chromosome. And you may know this, but males are more likely to be colorblind for red-green than females. And that's actually because it's on the X chromosome. And so we're going to talk about what that means. Okay. But here's just a few different genetic diseases that we've talked about. I skipped over Huntington's. It's very interesting, but we are going to practice it more in class just because this isn't about disease. This is more about the genetics. And then what caused the genetic disease. We will hopefully get a chance to watch this movie Gattaca, which kind of explores the ethics of what we call designer babies. So kind of like picking the baby that you want to have, whether that's a thing that we should be doing or should be even possible. And so I just want you to start thinking about this in the context of the real world, because there's a lot going on in bioethics right now that is questionable. And so people should be questioning. So with that, that's going to finish our lecture on genetics. And I will see you for non-Mendelian genetics next. So those are the things that don't follow Mendel's rules.